Okay, yeah, hello everybody. Thank you for the introduction. Thank you for the opportunity to speak here on the Allotrope Connect. Okay, could you go to the first slide? Or to the first slide after the main?
Michael, you should be able to control the slides yourself. There you go.
I control them. Oh, perfect. I think, yeah, okay. Yeah, okay, my name is Michael Rosskopf. I work in an IT department, work closely together with the R&D. And my role here is to coordinate allotrope activities at Bayer. I will give an introduction, show a bit our vision, explain a bit what this subtitle of our talk means, harvesting the digital dividends. But I'm not alone here. I'm together with two colleagues, with Schuette. He's a solution engineer from pharmaceuticals, and with Henning Kayser. Yeah, many of you will know him from previous talks here in this Allotrope Connect. He's the digital transformation lead from the crop science division. And later on, I will hand over to them, and they will give some more details on allotrope activities in the divisions. On the next slide, instead of something's coming. Thank you. Yeah, instead of starting with allotrope, we like to start with an analogy, which is related to music. That's maybe interesting because I learned when I started to work on allotrope topics that when you have started here years ago, then in the early days, it was a goal to develop something like an MP3 format for scientific data. That's not where we want to focus here. It's more about the process to compose music, and that's shown here on the left-hand side. Yeah, so there's in this example, a famous composer. He writes the notes, and writes the notes for all the different instruments. They are then arranged. Maybe things are tested on single instruments, and in the end, everything is put together. And then you need a symphonic orchestra to play it. And nowadays, the general process is, I would say, it's similar. You have lots of help today, maybe from different computer systems, for example. If you make electronic systems, electronic music, you would also create the different tracks of your song. You can directly listen to it, but it's an iterative process where you listen, where you add something and so on. And in the end, you have something you maybe had in mind before. And that's typically the way to make music. If you go one step further. Okay, here on the right-hand side, here's an alternative way to make music. Maybe you have heard of this. It's OpenAI Musenet, and this is something where music is created by an artificial intelligence. In more detail, it's a deep neural network, and it bases on a database with a few 100,000 songs that are there in a digital format. And what you can do is you can say, okay, create something that is similar, like let's say 9 Inch Nails and Mozart, if you like. By the way, that would be an interesting mixture. So it generates then from some, let's say, historic data, something new. This is, of course, just an example. Maybe many artists wouldn't use such a tool because it's a creative process. But if you have something in mind, you can come much faster, much closer to that what you want to, what you need. Of course, then you can edit it, you can extend it, and so on. But you would be a step ahead with such a method. And this is now then a good point to now go to a topic which is much more relevant for us here. That's the process to develop a new medicine here in this example. This is an example that's about 120 years ago. So there's a smart person, or in general, it's a team of smart persons, and they make lots of experiments. They use their knowledge, they use the state of research, and they generate lots of data. They will also have many failed attempts, of course, in order to develop something. And then sometimes in the end, there's a patent, and then a new medicine will come to the market. Let's assume, you can use all this data that has been generated before for a future development of something new. And for this approach, there's this little example here on this right-hand side. The prerequisite is that you describe your data in a good way, that you have a kind of a standardized format that is readable by machines. And here in this example below, this is just some kind of placeholder for typical methods and languages and so on that are used here for doing data science and analytics. If you then create something smart, then you can also be here a step ahead in this development. And you can reach the goal to develop something specific, some specific active pharmaceutical ingredients much faster. So that's a goal. I think this is very important for many companies, especially in these times of digital transformation. And that's what we name here harvesting the digital dividends. So if we can bring everything into such a smart format and use some smart methods, then we can be a step ahead. And this is, in fact, the vision we have. So on the next slide, question is now how to reach this. So of course, many things are necessary for this, and we cannot explain all of them here for sure. But one thing which is very important, and this is also not new to you, I think, is to make the data fair, means findable, accessible, interoperable, and reusable. And we think that the Allotrope Framework is a very important pillar to achieve this. And this is maybe then a second meaning for harvesting the digital dividend. So we and many other companies has now invested a long time into this Allotrope topic, and it has grown. We have now a matured format, for example, with ADF. And now we can roll out this on a broad scale. And let me quickly summarize the advantages. Of course, you will then have, if you do it in the right way, explicit knowledge, because you have semantic information attached to your data. If you use it, it will avoid that you lock up data in device-specific systems or LIMS systems, for example. And in the end, it will also improve how you can use data for data science, because then it's also very important that you have well annotated data. And another side effect, let's say, and that's very important for our regulated industry, is that it allows a clean and compliant archiving. But how reach this? How will it influence the landscape? And for this, I'd like to show you first maybe the typical current IT landscape here in a lab environment. What you can see here on the left-hand side, that's the side of the data production. You have lab devices with instrument control software. Maybe you have partnering companies, analytical clinical research organizations that produce data. And maybe sometimes you use also data that is generated on devices you not own, you have leased, and maybe they're exchanged every few years. And then this data is generated, and then it's submitted to other systems, like specialized systems, like chromatography data systems. If you have very standardized sample-based workflows, you will typically use a LIMS system. If it's more research-driven, then you might have an electronic lab notebook. In some cases, you combine these different types of systems. In the end, there is an archive where you store your information then for long-term preservation. And you might have a data lake for data science to do these things mentioned a few slides before. And you see here a very complex landscape, because you see all these different arrows, and every arrow requires in the end two interfaces that needs to be maintained. And another thing, which is also a big problem then for your data science, with every layer in between, you will typically lose some data. Because in a straightforward case, you maybe need just a few information for your typical straightforward workflow, but not everything you have generated here at the beginning. Of course, you can also add another arrow directly to your data lake here with lab devices, but that would only lead to much more interfaces, because if you have many different lab devices, that will end up in many additional interfaces. So what's the answer to this? And switch to the next slide. That's listed here. And to be honest, the main message of this slide here is, I guess it's very easy to guess, it's standardization. So with the Allotrope framework, there's a chance to standardize the output format of laboratory devices, standardize the exchange format for transfer between different systems, different applications, which is then also, you also will have then a standardized input format for your LIMS systems, your electronic lab notebooks, your archive, and also maybe if you use a data lake. If you have a data lake, you might typically map the data to other structures. Maybe you have some special databases in it, but you need some input format, and you also want to have here a standardized one. So it seems that Allotrope Framework is more or less in the center of all these things. And on the last slide, it shows a bit how to allotroperize the IT landscape and make use of it on a broad scale. And this is where we think that's the right direction. We again have on the left-hand side our data producer, but they can speak now the allotrope format. They produce the raw data, sometimes also the process data, and all these information enters a data access layer. And all the different systems are then attached to it. They're connected to it. And it's also possible, this is shown here in this example of the chromatography data system here at the top, to enrich this information in your ADF files. For example, if you make an analysis, you can write it back, you can update, and then all your different applications will benefit from it. This is, of course, a very high-level view, and I will now hand over to Eric, and he will give them some insights here.
So first of all, good morning, good afternoon, everyone. I hope you can hear me well. Please, Michael, you could confirm if you can hear me. Can you hear me?
Yes, I can hear you.
All right. Perfect. Thank you very much. So, great speaking to you. For me, it's the first time actually being here in Allotrope Connect as a speaker. Former times I was only listening. So my name is Eric Schuette. As Michael already introduced, I'm from our pharmaceuticals division out of the Chemical and Pharmaceutical Development Organization. So actually, we're the ones turning molecules which are already proven into then real drugs that you can take. And we are looking into the whole topic of allotrope since 1.5 years already, really proactively. Our organization is following our internal ambitions since many years, but we just started on really actively leveraging this Allotrope framework here in our organization. And if you could skip to the next slide, please, Michael. I would quickly like to explain what we are working towards. So we are having our chemical and pharmaceutical development analytical labs. So mainly those are labs dealing with method development, but also then with actually analytics for the things we are doing, for the formulations we are producing, or for the synthesis of pharmaceutical ingredients we are doing. And the majority of the work is happening under GMP regulations. So just to make you aware of the circumstances. And we have a quite diverse landscape of devices and also then of software and measurement techniques. To sum it up, 80% actually is HPLC GC, where we have a central mPower instance in place. We have mass spectrometry. We also have optical spectroscopy. That's a hard word for a German person here in such a panel, but I hope you understand what I mean. But besides, of course, we have balances, we have hardness testers, we have pH meters and so on. But actually, I just listed here the main technologies and associated softwares. And our target would be to get an allotrope file which fulfills all the requirements we have out of those main softwares from those main instruments. And then we accomplished in the past the implementation of a central lab system, which is called FELD, and which actually consists out of an ELN by BioVia and out of a LIMS system. And we want in the future to we want Allotrope to be actually the standard data format to get data into that system, which could mean extracting really results out of files and store them, for example, on the experiment base in some ELN documents, but which also means actually storing, attaching real ADF files to entries in the LIMS. So that is our central platform, and we want to have both the whole ADF and also the information out of the ADF files. And then we want to be able, for example, in a review, to be able to see our files, our results without going into the source system. And therefore, ADF is pretty helpful actually if you're just in the LIMS and then open the respective ADFs, and then you don't need to go to all those three source systems where you might have data from or even more, but you only have a central storage of all those files, and you're able to at least have a sufficient insight into what has happened there. And then based on out of that central lab system, we are also looking into digitalization initiatives, of course, as many companies are doing at the moment, which could happen, for example, with Python, with Jupyter notebooks, but these are just examples. And our target is then also to use all the data we have into our central lab systems. We might also need some data lake storage for specific use cases where we have a maybe large amount of data and where also those ADFs could be filled into. And yeah, actually, we want to then leverage this treasure of data which we achieve, which we get from our raw data, with those initiatives yeah, from a central storage like our central lab system. In addition to that, we're also having and many of you might know that if you're working under GMP, we need to archive our data, and that could be timeframes up to 30 years, depending on the data that what we are talking about. And there we are also currently looking into a Sonte space-based archive solution, which is also a central system here at Bayer, and which actually leverages ADF as a underlying format for the for the archive. And that could also be a potential storage which will become then relevant for those digitalization initiatives, where we focus on things like shortening development time, making processes more efficient, making less experiment by really choosing the real lab experiments we are doing based on our historical data, and having a standardized data format like Allotrope implemented here in our organization. That will really be a big step towards those ambitions. And with that, yeah, I'm done with my only slide, actually, I'm showing to you, and would be happy handing over to Henning.
Yeah, thank you, Eric. Yeah, and so I'm happy to share you the perspective also that we have at crop protection in the crop science division, in particular in the analytical lab environment, and would like to start, if you could switch, Michael, to the next slide, please, with like a general analysis where we are coming from. If you look at the left side of the slide, this is a like a very common architecture that you find in our environment. Just the department I'm responsible for deals with more than 100 different IT tools, scientific IT tools, to serve our scientific analytics, controlling instruments, providing data to different customer groups. So we have various LIMS systems that we're interacting with, various ELNs, and high two-digit number of instrument control systems. And with the desire to answer questions from our data that already came up in the past, we developed a lot of bilateral interfaces to that. So like, yes, I want to have within my chromatographic data system maybe some information from the balance and some sample information, and these were all linked. That led to a structure, as you see there, that is basically a gridlock situation of having huge number of interfaces that is very difficult to manage. And at the same time, it also keeps raw data locked in these source systems. So that's why we call them data hostage applications as well, because you never actually transfer and give access to the raw data to other downstream applications. If you report something, a result to a LIMS, you only do like a certain fraction, a certain content of a sample, API content, or you report some integrations, etc. So tabular values, but the raw data are usually locked in the source system. When we look into how we want to transform our organization and the way of working, as Michael pointed out in the beginning, how we want to generate ideation from our data, we need to change the structure fundamentally. This is not just from a data architect change, but also mindset change, how we want to organize the data in there. And so the first steps, if you conceptually what you look is, we need something like a transfer, transfer package that always describes the metadata and the context of certain scientific information and having a fair instance of raw and processed data, and making this the default transfer package to get out of this gridlock situation. And we are actually starting on that, on having go lives with mass spec and HPLC data in this scope, and using the ADF format, which is basically an instance of this concept, to central locations, and then enabling this to get displayed and analyzed from different angles for the workflow, actually from LIMS system, but also for further downstream analytics from data science applications and Python R applications, etc. So we can separate data from apps, which is very important to get there, and streamline on the same way also the interfacing. And just from this conceptual slide, I would like to show you what is already there. Michael, if you could go to the next slide, please. Because we already have a GLP validated Allotrope-based workflow already in place with mass spec data from SciEx Analyst and Thermo Excalibur, and we'll extend that with Thermo Chameleon as well. Going to something like central data access, and you see the different symbols that are symbolizing the raw data, that's the blue part, and the processed data that comes out of an application. For instance, that's the orange symbol in there. And so the data can flow between these different applications. So sometimes if you look at the LIMS, you don't want to have the raw data in the LIMS, so you just extract the information that is important there. This is done in human safety and residue analytics labs with a star LIMS-based LIMS system that actually reads from mass spec ADF files, the relevant part, and can be then further enriched and taken to the also ADF-based into that way. On the way, also the ADF Explorer is a crucial part to have a low-level and low-tech way of enabling users to just see what is actually in the files. And I just want to give you just a spotlight on one aspect of that. How do we use the Allotrope data files that we generate from these existing solutions? So you can all, the solutions mentioned there, you can take them more or less off the shelf, plug them in and install them, and there you go. How we bridge there the gap towards data science and data analytics, which in Python. And so on the next slide, I just want to show you an example how easy it is with just a few lines of code and the HDF5 Python library, how to read in I think in that case, I think a couple of thousands of chromatograms from injections from Chameleon in there, and just loading them into a Python class and variables, so you can use the common data analytic tools and what you actually would use to query the data, do inverse searches, whatever you might think of. We can do that with the mass spec thing as well. Even as you can see, if you want to reintegrate or finding peaks, allocating them in there. And this is basically the access how we use and enable the use of actually raw data in more data science applications. So on purpose, I'm not going any further deeper into this, because we have lots of ideas what kind of models we want to base and what we want to build on our rich analytical data, but we consider this as a competitive advantage, what we have developed there and what we are developing. It's just enabled by Allotrope, and which is the pre-competitive part. And but we have lots of ideas, and it can only inspire you also leveraging this potential that you potentially have also in your company. And so with that, on the next slide, I just want to highlight what actually Allotrope drives for us and enables us. So the Allotrope Foundation part is obviously enabling data verification for us, in particular in the analytical labs. And so supporting deep data discovery so get more out of the data that we already have and let us harvest that. And so it's also the basis, of course, for method repository delineation for workflow. So going back and get more asking questions that are otherwise difficult to answer with of the software, and changing, also enabling the change how a lab technician or a lab head will actually interact with data and work with data, and also providing these interoperable data sets as an open source, scalable, low-tech industry standard. So low-tech means it works for every lab, as it's a file-based solution. There are way more fancier ways of integrating things. But if you have a CRO or university that you collaborate with, this is still a very flexible, adaptable solution, just plug and play. And we at Bayer try to build on top of that, first harvest the digital dividends of our investments that we did in the past year. But now we are really at the stage where there's a lot of really readily available off-the-shelf solutions for the main CDS systems or mass spec systems that you can just install, plug and play LIMS systems and archives, and like harvest the dividends and generate benefits for today. And with that, I'd like to end and see if I'd like to answer any questions if there are any.
Great, thanks. Vinny, do you want to lead us through the questions?
Sure, I'll try. First check. How's my audio? Has my audio cleared up?
Sounds much better.
Yeah. Sorry about that before, folks. I found out afterwards I had bad audio, so I'll have to do the intro again next session. So anyway, thanks, Michael, Henning, Eric. Very, very interesting. Really appreciate seeing all parts of Allotrope being used in all parts of your infrastructure. Really cool. Just one question about some of the features that both in the pharmaceutical and the crop sciences that you had centralized the LIMS and Ellen systems. I saw definitely Allotrope format being used to send information into those applications. Are you also using Allotrope or Allotrope inspired terminologies and data models to structure the information in your LIMS and Ellen as well, and expanding beyond the instrument domain?
Maybe I take this first, and then Eric, you can add on that. So in general, with the approach that we started, we are somehow the structure of the data and the information that gets into the Allotrope data format is basically formed by the application that generate them. So some of those we work together, for instance, the Cihangir solution, bilateral approach, but hopped onto the other commercially available solutions there as well. So this is how the structure of the data is coming into the system. And of course, we are harvesting, in particular, when you come to the downstream solutions like the archiving, we are building on top of that structure and indexing the relevant parts for search purpose and structuring as well, and in a defined way. I think when it comes to LIMS, we are more still in a mapping space, because also that I think there's a technology gap between how the ontology that's usually used in the ADF and how LIMS master data and metadata sets are structured. So this is more low-level mapping exercise, but this is how actually we try to leverage the structuring of the information. Eric, if there's things to add.
Yeah, so you already covered half of my answer, actually. So also our LIMS solution is technically not ready to fully leverage the ADF ontologies and the semantics out of the box, but we are also looking into the topic of semantic data, and we are also building then own ontologies about the data we are having in those central systems, and therefore we are also using the Allotrope ontologies which are available to part of the data which might be into those systems as well. So yeah, we try to leverage that as much as possible, but that's just our systems were not or are actually too old to be out of the box compatible.
Yeah, understood. Thank you. Thank you very much.